right, guys, welcome back to part two of biochem. Um, we're going to talk here about the four different biomolecules, which you might remember are carbohydrates, lipids, proteins, and nucleic acids. I'm going to go in that order. Um, I really have enjoyed this stuff the more I've learned about it. So if you find that it's like kind of boring, give it a chance and try to think about it as instead of just like boring molecules, actually like the food that you eat and how it's sustaining you in a really cool way. So try to just kind of shift your thinking about it, especially when we're talking about structures and stuff like that, um, and see if that helps any. Okay, so we're gonna start by talking about carbohydrates, which are literally my favorite foods ever. Um, you know them as sugars and starches, including like bread, potatoes. Those would be like your complex carbs, and your simple carbs are things like sugars, simple sugars. So up at the top, I have labeled them as your sugars being your mono or your disaccharides. Anytime you see saccharide, that means you're talking about a sugar. So immediately, as soon as you see saccharide, you're like, yes, this is a carb. Got it. The other way to easily identify your carbs is that they will usually end with O's, usually as in almost always. But glucose, sucrose, fructose, all the O's, they're automatically carbs if you're trying to identify what they are. So two examples of sugars. Your most famous sugar is glucose. And then sucrose is a disaccharide. That's actually like the sugar that you use to bake a cake. That's like the simple sugar. Mono means one, like we talked about in the last video. Di means two. So if you have a disaccharide like sucrose, that's actually just two glucoses that are bonded together. They would have got bonded together using dehydration synthesis. And once you have two carbs that are bonded together, it's called a glycosidic linkage. Which is a covalent bond. So it's strong. Another hint that something you're talking about is a sugar is glyco. I'll just put that here, that glyco is also going to be sugar related. So saccharide, glyco, or ose, you're like, yeah, this is a carb, which should be kind of like a little helpful hint for you. Okay, simple sugars are used as short-term energy sources for living things. That's their number one use. So short-term energy, um, which is why like you eat carbs before you're gonna do a big exercise, or if you need like a quick energy boost, you eat like a candy bar, a protein bar. Oh, not really protein bar, but those protein bars that we like call them protein bars, they are high in protein, but actually what's giving you the energy is the sugar. So it's like a common misconception about proteins and energy. Um, Proteins as a food is not an energy source. Your carbs and your fats are energy sources, which we'll talk about um, in a few minutes. Okay, so moving on from that, starches are your polysaccharides, and you also probably have heard of them being called your complex carbs. Poly means many, so it's just many monosaccharides being linked together into huge, 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 huge chains, like thousands of sugars long. They're really big molecules. Starches can be used for short-term energy storage or for structure. So this is like short-term, relatively speaking, because fats are going to be your long-term energy storage. Um, Even a starch for us, humans do have starch in us that we like use to store our sugars, but it is still going to be short-term compared to a fat. So all of your glycogen, which is your animal starch in your liver, it would last you about 24 hours if you stopped eating. So it's still like a pretty short term when we're thinking about like energy and being sustained. So for animals, our storage starch is called glycogen. So that's an example of one sugar that does not end with O's, but it has glyco. So you should still be like, oh, yep, got it. It's a carb. It's stored in your liver. And what happens is when your blood sugar gets low, you have hormones that actually trigger your liver to release your glycogen so that it can be used to stabilize your blood sugar. Same thing when your blood sugar gets too high, you have a hormone called insulin, unless you have diabetes, that tells your liver it's time to take up some of that sugar and regulates it for you. So glycogen is really important for metabolism and blood sugar stability. For structure, we have another carbohydrate that humans don't use it, but animals like crabs, lobsters, and insects use it, and it's called chitin. It's used for exoskeletons, and actually fungi use it too in their like fruiting bodies, aka mushrooms. Plants also use starches for both energy and structure, but they use different ones. So for plants, their energy comes from a starch that's called amylose or amylopectin. And that's plant starch. Usually we just call it starch. So that's what you'll see it called a lot of times. And for structure, they use a carb called cellulose, and that makes up their cell walls. So, like, crazy thing about this is that humans can break down amylose, plant starch, but we can't break down cellulose, even though they're both chains of glucose. Because of the way the glucose molecules are orientated and bonded together, it actually causes our enzymes to only be able to break down one of them because of the way the molecules are actually shaped. So that's this note down here that talks about differences between starches have to do with the glycosidic linkages and the degrees of branching. That actually leads to us not being able to digest cellulose, which is why it is also known as plant fiber. So it's just kind of an interesting fact. I'm not going to go into all the details of like why it is that way. But if you are interested, you can look it up. It has to do with like alpha glucose and beta glucose and like the bonds flipping the glucose monomers actually like upside down in one of them. So, anyways, gonna move on from that. Carbs, super important. Big carbs, your polysaccharides, are really important for both energy and structure in both plants and animals. And your sugars, your simple carbs, are important for short term, super quick. Okay, here are just some examples of monosaccharides. So these are all simple sugars. They vary in size. And probably the most important thing that I want to make sure I say about this is that sugars are named for the number of carbons that they have. You'll see up here at the top that these two sugars are called trioses, which is my highlighter. And they're called trioses because they have 3 carbons. Pentoses are 5 carbons. So think about like a pentagon. Really important 5 carbon sugars are ribose and deoxyribose. Those are important for your DNA and your RNA. And then hexoses, like a hexagon, are 6 carbon sugars. Glucose is a 6 carbon sugar. Fructose is a 6 carbon sugar. And glucose and fructose, you can see actually on this image, are structural isomers. So they have different functions and slightly different properties, even though they're both C6H12O6, exactly the same. But because of the way they're laid out, they're actually different sugars. Kind of cool. Your hexoses, your glucose, can be formed into a ring also, which we'll model this week in class. And so can your pentoses. All right, gonna move on. All of these, sorry, I said I was gonna move on, but I'm actually not going to yet. All of these are monosaccharides, which means one sugar. So these are all simple sugars. None of them are starches. Starches would be like thousands of these things linked together into a massive, massive molecule, which I'll show you a picture of next. Okay, here is starch. So up at the top is plant starch. You have your amylose and your amylopectin. So again, they're really similar. They're just slightly different in the way that they branch and the way that they're bonded together, but they're both plant starch, and your body can break them down. The enzyme to break them down is actually in your saliva, which is why if you leave like a cracker in your mouth for a long time, it will eventually start to taste sweet. It's kind of gross, but you can actually do that experiment. And it's because these starches are made of sugars. So these little like hexagons are hexoses, 6 carbon sugars that are linked together into the starches. So when your saliva starts to break them down, you can start to taste the difference, that they're sweet. Glycogen is down here. This is your animal starch. That's what your body actually uses to store sugar for later when it needs it. So it's stored in your liver, but that's the difference between the two. Now, look at the difference in cellulose. So you can see that cellulose has a lot less branching. Here's your beta glucose that I was talking about. It's actually listed on this image, but it has to do with the fact that every time it's added to the chain, it flips upside down. In amylose, that doesn't happen. And because it's happening, it allows these hydrogen bonds to form, which creates a stronger, more stable structure. So the cellulose is able to form into these long strands. There's no branching, which forms microfibrils and a very thick and stable structure that your body's actually not able to break down. So that's the difference. Again, I'll just show your branching like starches up here, and then here's your cellulose. So just kind of going back to what I said in the last video about structure and function, going really, really, really hand in hand, like very much important that the shape is correct for the function of the molecule. Okay. Last carb that I want to talk about, which I kind of already did, is chitin. Chitin is a really strong structural carbohydrate for animals. So there's the monomer up there. Again, it's a polysaccharide, so it would be linked like thousands and thousands and thousands of these to make actually the exoskeleton of this cicada that's in the picture. But it's true for all arthropods. So like crabs use chitin, lobsters, spiders, all your arthropods, which arthropods are actually the most diverse phylum of animals, which is just a fun fact. Okay, let's talk about lipids. So lipids are fats. So in your examples, I'm going to put waxes, oils. Those are the ones that you eat, but they also are steroids, cholesterol, and some hormones. Not all hormones are lipids, but some of them are. So their functions, the main function that is going to be important for us is long-term energy storage, long-term energy. But we also talked about them being useful for insulation. So keeping animals warm, like whale blubber. Your fat is also really important for you staying warm. You just don't have as much of it as whales do. but it is really important. And then it's also important as signaling molecules in the form of hormones and important for making up your cell. Should have given myself more space here. Oh well. Lipids are nonpolar, which I might have talked about in the chemistry video a little bit, but they repel water. So they will not hydrogen bond, and they do kind of their own thing because of their nonpolar qualities. Since they're hydrocarbons, the hydrogen and the carbon pull about equally on electrons, which causes the bond to be nonpolar. So that gives them some of their properties that we actually care about. Typically, a lipid is formed into 3 fatty acid chains and 1 glycerol, which I'll show you on a slide in a couple minutes. But that's why they're typically called triglycerides, is because there's 3 fatty acid chains. Typically, we say that the monomer of lipids is a fatty acid. It's a little different than the other biomolecules, though, because it's not like a repeating chain of thousands of fatty acids the way that carbs was. It's going to be a glycerol and then 3 fatty acid chains. These 2 images here are showing 2 fatty acid chains and what they would look like. And I'll come back to that in a second. Another thing that's really important about lipids is that they make up phospholipids. Phospholipids is what makes up your cell membrane. So I'm going to put this here. You might remember them from freshman bio, but they have this like phosphate head and then these 2 lipid tails. Those are fatty acids. The top is a phosphate, so I'll just put a little P in there. That's why they're called phospholipids. And when I say it's a phosphate group, which is why we talked about those functional groups briefly in the last video. Okay, coming back around to saturated and unsaturated fats. So in a saturated fat, all of the bonds are going to be single bonds. And what happens is that causes the fat to not branch at all, which means what can happen is the different fatty acid chains can stack up one on top of the other and pack really, really close together, which makes them a solid at room temperature because they're more tightly packed. For unsaturated fats, they have double bonds or sometimes even triple bonds. The more double bonds they have, the more liquidy they're going to be at room temperature. So things like oils have more double bonds, which just means that the fatty acid chains can't stack as closely together. And that creates a more liquid consistency at room temperature. And you can see the difference in different oils. So olive oil is super unsaturated. Something like coconut oil, you can tell like is solid for longer than olive oil is. And that has to do with the double bonds in the molecules. So when we talk about trans fats, what happens with the trans fat is there is an unsaturated fat. It's super oily. People are like, this is annoying. I don't like it that it's oily. I don't like it that I have to stir my peanut butter before I eat it. It's really frustrating. I want it to be like smooth and creamy and delicious. So what happens is a process that creates these partially hydrogenated oils, which means that food scientists, go in, add hydrogens to the unsaturated fat to make it more solid. And that is a trans fat. What happens in trans fats, you can see in this picture that right now, this double bond, because look at where the hydrogens are. So this double bond right here, this is a cis bond because both the hydrogens are on the same side. If the hydrogen on the top here was down here on the bottom, it would be a trans bond because you would have this hydrogen, this diagonal line going across. It's going to change this branch of the molecule. So that means that the fatty acid chains will be able to stack a little bit more closely to one another, which will make them more solid. So that's why you're like Skippy peanut butter is like smooth and creamy, and your natural peanut butter is like stirred up because it's partially hydrogenated. Skippy. Okay, good. Glad we got that. Here's just a structure of a triglyceride. This picture is really horrible quality, but just wanted you to be able to see it, that your glycerol is in green, and then you have your 3 fatty acid chains. This would be this would be saturated because it's completely full with hydrogens as much as it could be. There's no branching in the molecules. So if it was room temperature, this would be a solid, like butter. Here is a diagram that just kind of shows your phospholipids. Phospholipids. We'll talk about more when we talk about the cell membrane, but what's happening here is that the fatty acid tails are hydrophobic, which that word might ring a bell to you. That means that they're water fearing. And it's because they're nonpolar. So because of that, they attract to one another. And then the hydrophilic, water-loving head is the phosphate. So this is why your cell membrane is a bilayer, because the fatty acid tails don't want to touch water. They want to touch each other, just like oil. So they create this double layer so that the fatty acid tails, which would be like these fingers, are together, and the phosphates, which would be like my hands up here, down here, are closer to the water. So it's a property of lipids that's really important for creating boundaries between your cells. All right. Last thing is just about trans fats. I have this here for you, but it's kind of what I just talked about related to what trans fats actually do in your body. You have good cholesterols and bad cholesterols, which I'm sure you have been familiar with before, probably. Your good cholesterols are your HDLs, your high density lipoproteins, which take your cholesterol and transport it back to the liver so that it can be processed. When you eat trans fats, it lowers the body's HDL levels. So it lowers your levels of good cholesterol that you have. Your bad cholesterol is your low density lipoproteins, LDLs, which transports cholesterol around your body instead of moving it to the liver. So what happens then is your arteries start to build up with cholesterol, which obviously can lead to a stroke or a heart attack. So you want to keep your HDL levels high, and the way to do that is eat less trans fats. Trans fats have gotten like a terrible reputation recently. Like, a lot of times you'll look at food for this reason, like they should have a terrible reputation. But if you look on food packaging, it will often say no trans fats, which is good. Okay. Proteins. Let's talk about them. All right, so proteins are a little bit more complex than carbs and lipids because their structure is even more important for their function. Monomers of proteins are called amino acids. And that's related to the fact that they have amino groups. So if you remember your functional groups, you have an amino group right here, amino, and you have a carboxyl group right here, carboxyl. Your R group is what's really important to the amino acid having specific properties. So this changes in every amino acid. It could be just a hydrogen, it could be a super long chain, it could be positively charged, it could be negatively charged. Yeah, and we'll talk about it when we talk about folding. There are 20 different amino acids, so that means there's 20 different R groups. That's it. I'll get them. These 20 amino acids combined together create every protein that has ever existed, which is amazing because there are many different proteins that do many different things. And proteins are kind of like if there was one molecule that like created life most importantly, besides DNA, I guess, it would be proteins because they do so many things. They digest your food, they signal around the body, they're hormones. Um, they're antibodies, they keep you healthy. They are doing so many things for your body constantly. Um, they are your traits. Whatever you see on someone is their proteins being expressed. So, like your skin color, your hair color, the texture of your hair, um, all of those things are related to the proteins that are being coded for by your DNA. And they're all happening just because of tiny little changes in the amino acid sequence of your proteins, which is amazing. Um, okay, I'm going to go down here to the bond type. So the bond type in protein is called peptide bond. It's still just a covalent bond. It's always done in the same order, though. So the carboxyl group of one will always bond to the amino group of the other. So again, here's the carboxyl, and here's the amino. The start of your protein chain will always be an amino. So here's the start at this amino group, and here's the end at this carboxyl group. And it will always be that way, that the start is an amino and the end is a carboxyl. In the middle are the R groups. In this situation, they just made both of them be hydrogen. This is a situation where dehydration synthesis leads to a nitrogen being bonded to a carbon. So the carbon of the carboxyl group of amino acid number 1 is going to be bonded to the amino group of amino acid number 2. When this happens, you create a chain that's called a polypeptide. So this is a chain of amino acids. A polypeptide is actually a different thing from a protein. So a polypeptide is just a chain of amino acids that have been strung together. A protein is a fully functional 3D folded polypeptide. So the polypeptide is like a piece of yarn. The protein is like a sweater that has many polypeptides together that have been maneuvered in the correct way to do a certain job. That's the difference. Functions I already talked about a bit, but there are so many. So digestion, that's enzymes, um, structure. Those would be things like keratin for your hair and your nails, collagen for like elasticity of your cells and your tissues. Those things are structural. Immunity, those are things like your antibodies, they're proteins. Cell signaling, those are going to be things like hormones. So insulin, like I was talking about before with carbs, insulin is a protein hormone. There are more. I'm just going to leave it at that for this moment, but there are more. Here's just a picture of the 20 amino acids. You will not have to memorize them in this class. If you go to med school, you will have to memorize them, but you will not have to memorize them for this class. So you can just see that there are different properties of them depending on the R chain. On all of these, the R chain is descending downward from the amino acid. So it's this, same thing on all of them. It's going down. You can see that some of them are way bigger than others. And this chart also has them labeled by polar or nonpolar or special. So that's going to be important for when we fold the proteins in a couple minutes. The R groups and their properties are really important for their ability to fold in the correct way. Okay, now let's talk about protein folding. So how a protein works is determined by its shape. That probably should be familiar now that I've talked about structure and function so much. So the shape is absolutely crucial to the protein working. The shape being right is dependent on this right sequence of amino acids being added to the chain. So before I go any further, I do want to say this, that when you eat a protein, your body digests it using enzymes, which are proteins. Your body digests it. And then the amino acids of whatever protein you ate are used to build your new proteins that your body needed. So used to build your proteins, used to build your enzymes, your antibodies, your hormones, all these different things. The order of the amino acids is determined by your DNA code. That's what decides it. So, super important that you eat proteins so that you can get the essential amino acids that your body doesn't naturally produce to build the hormones that your body like actually needs. So, just wanted to say that. Okay. So, the primary structure is just the string of amino acids. It's read from the amino end to the carboxyl, like I said before. So you can see in this diagram that here's your amino group and here's your carboxyl group there. Okay. That's the primary structure. It's literally just a string. The secondary structure happens when the primary structure, starts to fold up. So what happens here is the carboxyl groups and the amino groups are interacting with one another to form either a helix, which is called an alpha helix, or alpha helices if it's plural, or pleated sheets, which are called beta sheets. So this is done because between carboxyl and amino of the amino. And it's fun. So you can see that here in this diagram that the dotted lines are what's forming the helix or the sheet, and the dotted lines represent hydrogen bonds. So pretty weak interactions. They're holding these things together. Also, just want to say, I think this is probably clear, but the hydrogen bonds are happening between non-adjacent amino acids because they're already covalently bonded if they're adjacent. So it's going across or it's going like up and down. If it's twisted up like a spiral staircase, they won't be actually next. So that's secondary structure. Tertiary structure or third structure is folding up of the molecule because of the R group. So the secondary structure was related to the carboxyl and the amino groups. The tertiary structure is related to the R group. This is arguably the most important step because if you have an R group amino acid that's in the wrong place, it can literally affect the entire folding of the whole protein because let's say it's nonpolar and the other ones around it are polar. Now it won't fold right because of the interactions. So this is really important to getting your protein to have the correct 3D structure so that it can do its correct job. So at this point, you're going to have interactions that can be hydrogen bonds. They can be ionic bonds if your R groups were positively or negatively charged. They can be van der Waals interactions. They can be this disulfide bridge. This is really strong. This is covalent. This only happens when there's two sulfur groups. And then there's also this hydrophobic clustering, which is like what I showed on the phospholipids if they'll group together. That's your tertiary structure. Once you finish that, you'll have a 3D protein. Sometimes you will have multiple proteins interact together to form a quaternary structure, the fourth level. So that means you'll have multiple folded polypeptides that are connecting together to create an even bigger structure. You don't have to have a quaternary structure. Not every protein does, but a lot of them do. One, example of that is hemoglobin. So hemoglobin has 4 chains of folded up polypeptides and then iron groups, which are called heme groups, embedded in it. So it's a really big protein. But if it's got the wrong order of amino acids in the primary structure, even if one's wrong, it doesn't fold right. And people who have that have sickle cell anemia, which is a genetic disorder that like hugely affects their blood's ability to transport oxygen around their body. So really important. I can't say it enough. Shape and function. Shape, shape, shape. So important. Just want to say this too. We'll talk about it when we do enzymes, but high temperature, super salty situations and changes in pH, especially acidic pHs, can cause denaturing. Denaturing is unfolding. If it unfolds, guess what? It's the wrong shape. It can't do its job. It's done. So this is why it's really important to not have a super high fever for a super long time, because you can literally denature your proteins, and that's why you die from a fever. Also, just kind of cool fact that when you cook an egg, the reason it goes from being clear to being white is because you've actually denatured the proteins and they unfold, and that changes the properties of the egg white albumin protein and it makes it opaque. That's why when you cook an egg, you denature the proteins. Here's a picture of it. So you have step 1 up at the top, your string of amino acids is coded for by your DNA. Step 2, you have interactions between your carboxyl and amino groups, hydrogen bonds that form alpha helixes and beta sheets. You can have alpha helixes and beta sheets in the same protein. It happens. Your tertiary structure, you're going to get your 3D shape because your R groups are interacting. And your quaternary structure, you're going to get multiple polypeptides lumped together. So those are your 4 steps of protein folding. Now I think we're done with proteins. Yeah. All right, last thing. I know this video is so long. This is the last step. It's going to be pretty quick too. It's nucleic acids. We're going to come back to nucleic acids throughout the year, especially when we do like a whole unit on DNA. So I'm going to go pretty quick through it now. There are only 2 examples of nucleic acids that we care about. They are DNA and RNA. The D and the R tells you what kind of sugar they have. So RNA has ribose, DNA has deoxyribose, which just means it's the same as ribose, but it has no oxygen. Deoxy. Functions are that they are your genetic. So they're giant storage molecules. Think about them almost like a giant computer where the order of the bases in them is the code for the program. You're the program. Monomers of nucleic acids are called nucleotides. Nucleotides are made up of 3 parts: a phosphate group, a sugar, and a nitrogenous base. Your nitrogenous bases, there are 4 of them. Well, 5. But in DNA, there's 4. You have A and G, T and C, and then in RNA only, you have U. There are 2 types of nitrogenous bases. They're called purines and pyrimidines. The purines are a little bit larger than the pyrimidines. So this is important because a purine always goes with a pyrimidine. So that keeps the space in between the DNA the same. Because if it was like 2 small ones and then 2 big ones and then 2 small ones and then 2 big ones, it would create this like wavy ladder. We don't get that. We get a bigger one with a smaller one always. And that's why it stays like the same width. DNA and RNA are linear, so they twist, but they don't like fold up into a blob like a protein does. They always run in the direction of 3' to 5'. And when I'm talking about prime, that's related to the carbon. So because of the way that these guys are set up with their sugars, they have 5 carbon sugars, which means that each carbon can be numbered 1, 2, 3, 4, 5. The direction that's facing upwards tells you kind of the orientation. I'll show you a picture in a sec. So when you build a polymer of either DNA or RNA, you're going to add your nucleotide always to the 3' end. This is important for later on when we talk about how DNA replicates itself, because in DNA, the strands run anti-parallel, which is this opposite direction situation. So that means one is running in the direction of 3' to 5'. The other one is running 5' to 3'. So one of them has to be copied backwards because the nucleotides always get added to the 3' end. We'll talk about that more later on. Okay. So here's a picture of just our nucleotides and how they come together. They come together using hydrogen bonds, which you can see in this top picture. And just remember that T always goes with A and G always goes with C. So I usually tell my freshmen to remember that by at Cardinal Gibbons, ATCG is how they go together. Here's your diagram of your nucleotides. Obviously, this will be different up here, the nitrogenous base, depending on whether it's A, T, C, or G. Then you have your sugar here that helps you to determine which ends are 3' and which ends are 5'. So based on this up here, our 3' hydroxyl of the sugar is the end of the chain. The 5' phosphate is attached to the sugar. So down here, this is our 5' because it's attached to the phosphate. This is our 3'. So that means that new nucleotides would be added on to the 3' end. New nucleotides added here. And the 5' will always be the one that's attached to the phosphate. Here's just another little picture that compares DNA to RNA. Probably it's very familiar to you, but RNA is single-stranded and DNA is double-stranded. Then, of course, remember that DNA runs anti-parallel. So one of the strands is running in this direction and the other one is running in this direction. So basically, like one strand is upside down on DNA. And then also remember that uracil is found in RNA, whereas thymine or T is found in DNA. That's a big difference. Okay, last thing, I promise. There are some biochemicals that are combos of two different groups. Two examples of that are important for us are glycoproteins. Glycoproteins are important for the cell membrane. They are signaling molecules. So they are actually the reason that your blood type is whatever it is. So if you're A, it's because you have A glycoproteins on your red blood cells. If you're AB, you have both A and B. And if you're O, you have no glycoproteins. But it's basically just a protein with a sugar sticking off the top. Lipoproteins I talked about a little bit when we did lipids, but they are related to your cholesterol. So they're proteins that are also have some lipid parts to them, and those are hybrids. So that is possible too. Okay, guys, I know that was so long. Thank you so much. You're doing great. Let me know if you have questions and how I can help you. And that's it. Great job. Crushed it.